Hi, my name is Ken Valkenburg, and I'm a cancer researcher at Johns Hopkins. I'm going to continue this lecture series on understanding cancer metastasis that Dr. Kenneth Pienta introduced in Lecture 1. I'm going to talk to you about primary tumor growth and angiogenesis during the process of cancer metastasis. There are 4 objectives for this module. At the end of the module, you will be able to describe uncontrolled cell division, which is a defining property of cancer, define what a mutation is and describe how it plays a crucial role in the tumor formation, define neoangiogenesis and describe its role in tumor formation, and finally, describe a primary tumor microenvironment. There are many cell types in a tumor, not just tumor cells, and you'll be able to name several of them by the end of this lecture. Our first section will go over how a tumor is initially formed through uncontrolled cell division. I will define cell division and describe how a cell normally goes through this process as well as what goes wrong in cancer. First, I'd like to revisit the entire metastatic process with you, from primary tumor growth to metastatic tumor growth. I covered this process in the Introduction to Cancer Biology series of lectures, which can also be found on the Coursera website. I will briefly go over the process with you. The process of metastasis begins with the growth of the primary tumor, which will provide the necessary seeds or tumor cells for metastatic tumors. Primary tumors grow as a result of accumulated mutations in the DNA, and they expand via growth of new blood vessels, and this is known as neoangiogenesis. They undergo physical changes, primarily epithelial to mesenchymal transition, which allows them to become mobile. Tumor cells must then invade through the extracellular matrix, or ECM, and intravasate into blood vessels. Once in the blood vessel, cells must survive circulation and make their way to a secondary site, which is any other organ besides the organ from which the tumor cell originated. For instance, prostate tumor cells typically metastasize to bone as a secondary site. Tumor cells then extravasate out of the blood vessel and into the secondary site, where they may grow or undergo dormancy, which is basically just not growth. Over the course of the next several lectures, our group of instructors will go over each of these steps in detail to provide a much deeper level of understanding of the metastatic process. In this lecture, I will cover the first step, primary tumor growth, and the second step, angiogenesis. To start, I'd like to give you a quote from a very influential book about the history of cancer. And this book is called The Emperor of All Maladies. It's written by Siddhartha Mukherjee and published in 2010. And the quote is this: "Cancer is a disease caused by the uncontrolled growth of a single cell. This growth is unleashed by mutations, changes in DNA that specifically affect genes that incite unlimited cell growth. In a normal cell, powerful genetic circuits regulate cell division and cell death. But in a cancer cell, these circuits have been broken, unleashing a cell that cannot stop growing. That this seemingly simple mechanism, cell growth without barriers, can lie at the heart of this grotesque and multifaceted illness is a testament to the unfathomable power of cell growth." This quote gives you a sense of just how important cell division is in tumor development. In fact, cancer is defined by that very property, the ability of a single cell to divide continuously and uncontrollably. And depending on what that initial cell is, different types of cancer can arise. If an epithelial cell, which is the lining cell of most of the organs of our body, divides uncontrollably, the resulting tumor is referred to as a carcinoma. Carcinomas represent about 90% of all cancers and include the most common cancers in the world, which are prostate, lung, breast, kidney, colon, and stomach cancer. If the originating tumor cell is from a connective tissue, like muscle or bone, the resulting tumor is referred to as a sarcoma. Sarcomas are very rare and represent only about 2% of cancers. An example of this type of cancer is osteosarcoma, and about half of all osteosarcomas are diagnosed in children and teenagers. Finally, if the originating cancer cell is a type of blood or blood-forming cell, then the resulting cancer is a type of leukemia or lymphoma, and these represent about 8% of all cancers. So, what is cell division? Cell division is when one cell becomes 2 cells. In general, we think about cell division as producing 2 identical daughter cells, but as we will see later, that rule doesn't necessarily apply to cancer. The official term for cell division is mitosis, and you will likely hear me say that throughout this lecture. One thing of importance to note here is that the daughter cells can also divide, meaning that if every cell that is produced divides, there is an exponential increase in cells, so 1 becomes 2, which becomes 4, 8, 16, 32, 64, and so on. This gives a perspective on how powerful cell division can be. In the previous slide, I spoke in simple terms about cell division, 1 cell becoming 2. The reality is that cell division is highly controlled by regulatory cellular machinery. Cells go through several phases in what is called the cell cycle, which prepares the cell for division and performs a sort of quality control on the cell to make sure it's ready to divide. There are 4 main phases in the cell cycle. The first gap phase, or G1, also known as a growth phase, allows for the cell to synthesize proteins and grow in size. The next phase is the synthesis phase, during which all of the DNA in the cell is duplicated or synthesized. In order for the cell to split, it must place half of its DNA into each daughter cell. And in order for this to happen, the cell must double the amount of DNA, and the new DNA is identical to the existing DNA. This is one place where mutations in the DNA can occur if errors are made in the duplication process. G2 is the second gap phase, where the cell continues to grow. Cellular machinery, made out of specific types of proteins, will also double-check the DNA and make sure the cell is ready to divide. The final phase, or the M phase, is mitosis, which is the shortest phase in terms of time. Mitosis consists of prophase, metaphase, anaphase, and telophase, during which the cell splits all of its contents, including DNA, equally between 2 identical daughter cells. And then the cycle of the 2 daughter cells resumes with G1. The cell cycle also provides time for the cell to perform quality control on all of the cellular components to make sure the cell is healthy enough to divide. The goal of the cell cycle is to produce 2 viable, healthy daughter cells. So if the cell is damaged or unhealthy in any way, the cell cycle prevents division from taking place. It does this through 3 checkpoints throughout the cycle. The first checkpoint is in the G1 phase, called the G1S checkpoint. Cell checkpoint proteins check the cell for its size, access to nutrients and growth factors, and DNA damage. If everything is found to be satisfactory, the cell is allowed to proceed to the S phase. The G2M checkpoint similarly checks the cell for the appropriate size and ensures that the DNA has been duplicated correctly. And then finally, the spindle checkpoint makes sure that the chromosomes attach to the mitotic spindles correctly during mitosis. If they do not attach correctly, something called polysomy can occur, in which one of the daughter cells has more chromosomes than it should. An example of this is trisomy 21, which you may have heard of, because this causes Down syndrome. So if the checkpoints don't find anything wrong, the cell is allowed to pass the checkpoints and proceed through the cell cycle and is able to divide. If, however, some sort of damage, such as a DNA mutation, for instance, is found, the cell has 3 options. The damage can be repaired by cellular proteins. If it cannot be repaired, the cell may senesce, which basically means that the cell permanently stops dividing but is not dead. And the third option is that the cell may undergo programmed cell death. Programmed cell death is also known as apoptosis, which is Greek for falling off. It was coined by the original cell death researchers to represent a natural death of leaves, which then fall off of trees. As I've mentioned, when cellular damage like a mutation is found during the cell cycle, the cell may undergo programmed cell death. In this way, only healthy cells are allowed to divide and unhealthy cells are gotten rid of. If cells all over the body do this efficiently, this process promotes overall health of the cells, tissues, organs, and then therefore the organism as a whole. And in this diagram you see a healthy cell becoming 2 healthy daughter cells, and then one of those cells becoming a damaged cell, which then undergoes programmed cell death. In cancer, however, something goes wrong with this process of weeding out the bad cells. Damaged and mutated cells are allowed to divide. The cell loses the ability to control this division. How? 2 examples of how this can happen are as follows. Cell damage occurs, but the cell cycle checkpoint machinery is unable to detect the damage. Therefore, the damaged cell is able to slip through the checkpoints and successfully divide. Alternatively, the damage is detected, but the programmed cell death is halted for some reason. The cell death machinery is faulty. These things are likely due to mutations in the genes that control the cell cycle and in genes that promote rapid cell division, which can cause the cell to make mistakes and move through the checkpoints too quickly. We're going to cover mutations more in the next section. So once a damaged or mutated cell makes it through the cell cycle, it creates 2 damaged daughter cells, which then may create 4 damaged daughter cells, and so on and so forth until you have an entire tumor full of these damaged cells, and that's what the diagram represents. As we'll see later, these cells often gain further mutations, which turn them into even more aggressive cancer cells. So in this section so far, we've talked about how cells can become damaged and turn into cancer. Most of the damage we've been referring to is in the DNA, which resides in the nucleus. These cells become quite abnormal over time and are recognizable when placed under a microscope. In fact, to this day, the way cancer is largely diagnosed is by a pathologist studying the shape and the look of the cells taken from a patient. This diagram shows some of the things that pathologists look for when making a diagnosis of cancer. They study the amount of cytoplasm in a cell, and cytoplasm is all of the parts of the cell that surround the nucleus, so everything non-nucleus. Cancer cells tend to have a larger nucleus and smaller cytoplasm. Cancer cells can also have multiple nuclei in individual cells, whereas normal cells only have one nucleus. The chromatin, which is another word for DNA wrapped around proteins in the nucleus, also looks different between cancer cells and normal cells. Here is an example of normal prostate tissue, which is a very organized and confined structure. The cells line up in a row, and the nuclei, which are the purple dots in this image, look uniform throughout the tissue. Each cell has only one nucleus, and you can see a lot of the pink color, which represents the cytoplasm. In contrast, if you look at the image on the right, the prostate cancer tissue is very disorganized and has lost its uniform shape. The nuclei are large and look different from cell to cell, and you can also see very little cytoplasm, which again is the pink color. This gives you a little bit of an idea of what pathologists are looking for when they look at a tissue biopsy taken from a patient. So that concludes our section on uncontrolled cell division, and then we're going to get back to the lecture and go over mutations in DNA on the next section.